video is just to look at the genetic analysis chi-square homework to make sure that we know how to do it. Make sure that you're paying special attention to this. Come in if you need help, because I know it's kind of hard to wrap your brain around, but I wanted to make sure you have this to go back on to view in case you're someone who is struggling with this, is struggling, how do I figure this out? So I'll go through all 4 of these questions, and you can give me your feedback. So, number 1. You and a laboratory partner are conducting an experiment to see whether 2 genes in fruit flies are linked. You have noticed in previous studies that hairy legs are dominant over non-hairy legs, and that red eyes are dominant over white eyes. You cross one parental fly that is heterozygous for both traits with another that is homozygous recessive for both traits. So, big H will be a hairy legs, little h non-hairy legs, big R red eyes, little r white eyes. So these are the parental genotypes. Heterozygous for both, homozygous recessive. So I put the heterozygous for both along the top, homozygous recessive along the side. And since the homozygous recessive parent can only contribute a recessive allele for both genes, all 4 of their gametes are going to be the same, which means if we figure out one row, all the other 3 will be the same. So we can actually just cross out these other columns and just figure these out. And that's what you should get when you fill in the boxes. So we have 4 different genotypes and an equal probability that an offspring has either of any of the 4 genotypes. So when asked to predict what ratio of these phenotypes from your Punnett Square, each of them is just as likely, 25%, or the Mendelian ratio of 1 to 1 to 1 to 1. So when we look at our experimental data, we have what we actually observed. This is going to be my observed column, which I actually observed. What would we expect if this was a Mendelian inherited gene, so not linked? We would expect 1/4 or 1/4 of each of these, of 5,080 to be each of these genotypes. So that divided by 4 should be what we expect, and that's going to be 1,270 for each. Now we're going to calculate our chi-square. So chi-square, the sum of observed minus expected squared over expected. So I did a column, observed minus expected squared over expected. And here's roughly those numbers. Very big numbers. So when I add them all up, we get 3,350. Now, what we're expecting is that it is Mendelianly inherited. There is equal probability of the 4 phenotypes happening in an offspring. If it was linked with no crossover, we would expect half of them to be this phenotype, half of them to be this phenotype, and none to be this phenotype. Why? Because the ones that are the least common in our offspring are most likely our recombinants, where the crossing over happened. If there wasn't any crossing over, they would all be one or the other of these 2. So we're going to look at our chi-square table to tell us what 3,350 means. So there's 1, 2, 3, there is 4, 1, 2, 3, 4 possibilities. Our degrees of freedom are our number of possibilities minus 1, so that our degrees of freedom are 3. If you look in this table, under the 3rd column, 3,380 is way, way, way below a p-value of 0.01. That means that there is less than a 1% chance that the deviation in these numbers from our expected is just due to random chance. What does that mean? It means there is a 99% chance that this isn't Mendelianly inherited. There's a 99% chance that they are linked. 99% chance. So we're going to go down, and we're going to write in, use your Punnett Square to determine whether the hairy leg gene and the eye color gene are linked. Well, we can now be 99% certain that they are. If the genes are linked, predict which alleles are linked. Which alleles are linked? Well, they're going to be the alleles responsible for the non-recombinant phenotypes, the hairy legs and red eyes, and the non-hairy legs and white eyes. So, hairy legs is dominant H, red eyes is dominant R. They're in the same chromosome. Non-hairy legs, recessive H, and white eyes, recessive R, are on the same chromosome. So, we would suspect that the dominant H and the dominant R are on the same chromosome, and the recessive H and the recessive R are on the same chromosome from our data, because they are most often inherited in this order. Those individuals who get a recessive H get a recessive R, and those who get a dominant H most likely get a dominant R. The crossover products have mismatch, recessive R's with dominant H's and recessive H's with dominant R's. Now we're going to figure out, since they're linked, what the distance between the two are. So to do that, we're just going to do the number of recombinants divided by the total number times 100. So the number of recombinants, those two middle, is 480, and the total of all of them is 580, or 5080. So they are 9.4 map units apart, and that would be our answer. Alright, number 2. You and your professor are working on another genetics experiment. You're wondering whether a specific fruit fly trait wing length follows a simple Mendelian inheritance pattern of complete dominance. Long wings seem to be dominant over short wings. You breed parental fruit fly who is heterozygous for wing length and a homozygous recessive fruit fly. So here are their genotypes. Heterozygous, homozygous, recessive. And here's our Punnett Square. 50-50. Half of them have 50% probability of getting long wings, 50% probability of getting short wings. So, after breeding fruit flies, we're raising 100 offspring, and these are their phenotypes, and are observed and are expected. Now, we expect it to be Mendelianly inherited. We expect 50-50, 250 long wings, 250 short wings. When we do our chi-square observed minus expected squared over expected and add it up, we get a p-value of 6.272. We'll go to our chart. We have 1, 2 degrees of freedom. No, we have, 1, 2 possibility minus 1. We have 1 degree of freedom. So it's going to be in this column here on the table. What was our number again? 6.272. 6.272 is in between 3.84, a p-value of 0.5, and 6.64, a p-value of 0.01. What does that mean? There is in between a 95 and a 99% chance that this difference between observed and expected is due. There's only between a 0.5 or a 5% and a 1% chance. A 5% and a 1% chance for our p-value 0.055% and 1%. There's between a 5% and a 1% chance that this observed and expected is due to random chance. So there's between a 95 and a 99% chance that this isn't Mendelianly inherited, that something else is going on. Okay? Make a claim as to whether the wing length follows a simple Mendelian pattern of inheritance. We can say that there is between 95 and that it does not follow Mendelian pattern. not follow a Mendelian pattern. It's not close enough to 50-50 with our total sample size to be able to safe to say that. So in this case, we got something that doesn't follow. It's not quite there. But we can't say with a greater than 99% accuracy that it doesn't. Last, you and your laboratory assistant helping a professor research the inheritance pattern of specific phenotypic traits. Antenna length and eye color. In your research, you predicted that red eye color dominant R is dominant over white eye color recessive R, and long antenna dominant A is recessive over short antenna recessive A. We are testing our hypothesis that the genes are inherited in Mendelian pattern. We're making our test cross with two fruit flies that are both heterozygous for both traits. So those would be their genotypes, and when we cross them, we get this Punnett Square. And we're going to observe a classic Mendelian. We cross two heterozygotes. A classic Mendelian trait to give a ratio of 9 to 3 to 3 to 1. 9 dominant dominant, 3 dominant recessive or recessive dominant, and 1 recessive recessive. So we need to find if this follows a 9 to 3 to 3 to 1 ratio. Here is our data, and we'll see if this, using chi-square, follows a 9 to 3 to 3 to 1 ratio. So, what do we observe? This is our data from up there of our actual, what we observed, our expected. This is what 9/16 of 2000 would be. This is what 3/16 of 2000 would be, and 1/16 of 2000 would be. Alright? So our hypothesis is that this follows a Mendelian pattern of inheritance. We do our chi-square analysis. We get our number of 293.2 with 1, 2, 3, 4 possibilities, minus 1, 3 degrees of freedom. 3 degrees of freedom, and that's way off the end, so way less than 1% chance that our observed data differs from the expected by random chance. So we're going to reject this. There's no way that this is just a Mendelian pattern of inheritance. So we have a less than 1% chance that that the data is due to random chance, so we can be greater than 99% certain it is not Mendelian pattern. So they could be linked. They could be a linked gene. That might be what this is. Probably not. I don't see too clear linked or recombinants and too clear parentals, but hey, you never know. Last, given the crossover frequency of each of the genes in the chart, construct a chromosome map. Here's our crossover frequencies. We need to remember the greater the crossover frequency, the farther they are apart. So these equal map units. So we're going to construct a chromosome map. So we're going to construct a make-believe chromosome. And where are the genes? Is A here, is D here, is C here, and is B here? And we're going to do that by putting these genes on a chromosome until all the distances work out. And this is just kind of a puzzle, trial and error. And so I did that below. And now if I put them in this order, like this, B, A, D, C, it all works out. There's 15 map units between B and A. There's 25 between A and D. There's 30 between A and C. And that matches my percent recombination from up there. Alright, that's it. Please look over this corrector paper and let me know if you have any questions.